We'll welcome our next speaker, Dr. Shane Liddelow from New York University Grossman School of Medicine, and his talk's title is Reactive Astrocyte Responses to Inflammation and Neurodegeneration. Welcome.
But what I want to talk about today, unfortunately, is not going to touch on Down syndrome, so I apologize in advance for that. But what I really want to talk about is something that I think might be applicable to Down syndrome, although we haven't looked. Lots of other people who are doing really exciting science in that space definitely have looked. But what my lab is interested in is a very disease agnostic approach to looking at the responses of astrocytes in the brain, which we've heard about in the last few talks, and microglia, peripheral immune cells, and how those inflammatory responses are common to a whole host of neurodegenerative diseases. And today I'm going to focus more on Alzheimer's disease, but if anyone is interested in other diseases that you may be working on, I'm more than happy to talk about that in the question time or offline. What I would like to do, though, is really start by highlighting the people that have done the work that I'm going to tell you about today. The sort of spearhead of the vignettes I'll tell you were run really by amazing postdoc Jessica Sadik, who's now left and is the senior scientist at Valor Health, a graduate student in the lab, Michael O'Day, and another senior postdoc in the lab, Philip Hazel. And they were readily helped by all of the other people on the slide, and I'll also acknowledge them as we go throughout the presentation. So we had some really beautiful introductions to astrocytes and microglia, both yesterday from Michael and Beth, and also this morning from Nancy. And so I won't go into it with too much detail, but suffice it to say, my lab is really at its heart a cell biology lab. So we're very interested in how individual cells interact with one another in the brain, either in a normal homeostatic physiological state or in the context of a whole host of different pathologies. And so a few years ago, a graduate student in my lab, Rachel Kim, drew this beautiful wheel of astrocytic function that I think sort of gives us a very broad summary of exactly what we like to look at. And that is, we like to look at functions first and foremost. And so we ask very simple questions. Are there normal physiological functions that astrocytes could lose in the context of different neurodegenerative diseases? So for instance, if astrocytes lose the ability to provide trophic or metabolic support to neurons, would that manifest as an initiator or progressor of neurodegeneration in different diseases? We're also interested in whether or not astrocytes are still able to do brain building tasks like the really important synaptogenic function they have, and whether or not they would lose that could be really important in synapse density loss or inability for memory and learning to occur. Now, of course, maybe these astrocytes don't completely lose these functions. Maybe they just become a little less efficient at that. And so I just put a few examples here where, maybe it is not that astrocytes don't provide any metabolic support, but maybe they just provide a little less of that. And so neurons are no longer able to run at full efficiency, and that could also be problematic. And maybe these astrocytes are not as efficient at reuptake and recycling of neurotransmitters, and this could lead to things like excitotoxicity if there's excess glutamate in the synaptic cleft. Now, of course, there's also the possibility that the astrocytes don't lose any functions, but they could gain in functions that could be important in understanding the context of disease. And so one example is maybe these astrocytes, which are normally post-mitotic, could re-enter the cell cycle, become highly proliferative, and form what has been historically thought of as a scar or a border that could either corral sites of injury, like in a stroke, or could surround any particular pathology in the context of Alzheimer's disease that could be amyloid plaques. And then, of course, there's the possibility they could gain other functions. This is something that we've been working on for a number of years, and that is the fact that these astrocytes could gain a function that they don't normally have and could be active contributors to the degeneration. And that would be the idea, as Gil brought up just earlier, the idea that these astrocytes could be driven to a neurotoxic state by IL-1 alpha and TNF, and this could see the release of an active neurotoxic compound. I won't talk about that today, but we have spent a number of years showing that this is actually a toxic lipid that is released specifically by astrocytes. Now, needless to say, we don't like to suggest that any reactive astrocyte would have all of these functions lost or gained at the same time. They may have them, as we talked about earlier, that perhaps they have some functions early in diseases and other functions later in disease. And disentangling both those spatial and temporal dynamics is something that we've become progressively more interested in. So how do we do that? Well, of course, nowadays you do that with single-cell transcriptomics. It is expensive, but by per unit of data, it's relatively cheap. And this is something that Philip Harsel in my lab really set out to try and uncover a number of years ago by using single cell and trying to generate extremely large, powerful data sets of these astrocytes. So this is just a 3D UMAP, just to show that there is one moving image in my slides here today. But what Philip was actually interested in was the response of astrocytes, the sort of heterogeneity of response to astrocytes to a very basic inflammatory model. So he was using systemic injection of lipopolysaccharide, a bacterial cell or endotoxin. He was interested in whether or not there was differences in the responses of both female and male mice. And, you know, I won't go through all of this in depth, but suffice it to say, he sequenced around 80,000 astrocytes. He knew they were all astrocytes because they were expressing canonical astrocytic markers. And then he could do the stock standard analysis pipeline and say, are there specific markers of individual clusters of these astrocytes? And could that give us some interesting hints about what these astrocytes could actually be doing in the context of, in this case, a simple inflammatory insult? Now, we're very interested in all of these different subtypes, and we're going through them systematically, working out where they reside and what their functions are. But what I really want to focus on today is this one particular subtype of reactive astrocytes here. It's extremely lowly abundant. It represents only about 2 to 3% of all of the astrocytes in these inflammatory insults, and was also positioned at a very strategic location, which will be a theme we'll run through the rest of the vignettes of the talk now. So this particular subtype was very distinctly identified by a bunch of interferon responsive genes. And knowing that, Philip was able to use in situ hybridization to go and localize where these cells existed in the brain. And so here we just have some in situ. In red, we're showing a marker of these particular subtype of reactive astrocytes, an interferon response gene, IGTP. And we can see it's co-localized really nicely with this astrocytic marker GLAST, or SLC1A3, shown here in magenta. And what we can see is actually these astrocytes are present on the outer surface of the brain or in layer 1 of the cortex, and they have some penetrating processes going down along these vessels here. So the first question that Philip and I had was, well, is this actually astrocyte specific, or are there some endothelial cells that are also expressing this marker? And so he also did some co-localization of this interferon responsive reactive astrocyte marker with Claudin-5 here, a marker of endothelial cells, and showed that actually they weren't endothelial cells upregulating this gene. It really was specifically the astrocytes in that outer cortical layer. So we have these juxtavascular astrocytes that are upregulating interferon response genes that we had not seen previously. Now, of course, this is only a single marker, and we would rather prefer to identify astrocytes and show astrocyte location using a whole suite of different differentially expressed genes that Philip had identified using his single-cell transcriptomics. And so we started to make the shift from using single probe in situ hybridization to using spatial transcriptomics in a very unbiased way. And this brings up a first problem, that is that the resolution simply is not there to address the question at hand. And so this is just a very simple example of 10x visium sections here from a normal physiological brain section, mouse brain on the left-hand side, and an inflamed mouse brain on the right-hand side. And here what Philip has done has probed for a single marker of these interferon responsive gene astrocytes, this IGTP. And if you squint, you could argue that there's slightly more highly expressing spots on the right-hand side than left-hand side, but it's a hard argument to sell. And so what Philip decided to do was actually combine, rather than individual markers of these genes, to actually induce expression or show expression of a whole suite of genes, a module of these genes, up to 200 or so of them. And when he did this, he was able to see that there was actually some low-level hotspots on the outer surface of the brain, but also a striking hotspot of expression of all these genes collectively in the same region at this other strategic location, which is around the ventricular system. So we started building this hypothesis that these astrocytes were strategically located at ports of entry into the brain, and perhaps they were an interface between central nervous system immune cells, microglia, and peripheral immune cells that would be releasing the interferons. So another way to look at this was sort of spearheaded in the lab by Mike O'Day, And this was taking advantage of a Bayesian next nearest neighbor mathematical approach that was published by Ed Zhao in Nature Biotechnology last year. And using this, what we're able to do is essentially enhance the resolution of our spatial transcriptomic data. And when Michael did this, he could see when he uses a single gene here, this IGTP, rather than having a spotty image, he has a very distinct hotspot of expression around the ventricular system, but we couldn't detect the location of these cells on the outer surface of the brain. So he decided to combine both this base-based method with the module probing that Philip had done, and now we could actually see hotspots around the ventricular system, along the foramen connecting the ventricles, on the outer surface of the brain, and even tracking along this penetrating vessel here. So obviously this is nowhere near as high resolution as the in situ that we have, but it enables us now to very quickly localize substates of reactive astrocytes to different locations throughout the brain, so that when we then probe back in to do functional studies or to do in situ studies, we know exactly where we might be likely to look. So of course, that's all in mouse, and we would also like to see whether or not this is evolutionarily conserved as a response. So is this actually present in human patients? Because if it's not present in human patients, we're probably not going to spend a concerted amount of time developing therapeutics to try and modulate those cells' function. But luckily for us, Brad Hyman's group up at Mass General around 2 decades ago had actually published a paper in which they were looking at the response of microglia in Alzheimer's disease, and had stained the brains of these postmortem human tissues with an antibody to the chemokine CXCL10, which of course is expressed by microglia, but is also highly expressed by these interferon-responsive astrocytes. And if we look at these images here, we can see on the left-hand side there's a low level of this immunoreactivity, but on the right-hand side in these Alzheimer's disease patients, you can see there's a marked increase in not only the levels of that reactive protein inside the astrocytes, but also the numbers of astrocytes that are being induced to take on this reactive substate. And so Jessica Sadik in the lab decided to say, well, is that also true across a whole host of different Alzheimer's disease mice models? And unfortunately, what we found was that the single-cell transcriptomic data, while very high quality, was very lowly underpowered. And because we're looking at a substate of cells that is maybe only 2%, these reactive astrocytes had been missed. And so what Jessica did was she took advantage of a couple of these data sets and decided to enrich their power by integrating a whole host of data sets with Philip's large data set to see if she could see them. And so that's exactly what we have here in this TISNE. In blue, we have Philip's large inflammatory data set. In red and yellow, we have some data sets from Marco Colonna's lab using the 5xFAD mouse. And in purple, we have some data sets from Mikhail Schwartz's lab, also using the 5xFAD mouse. And if we focus on these data here from Marco's lab, we can see at 7 months in these 5xFAD mouse, there is a marked increase in the number of cells that are positive for these markers of these reactive astrocytes. I'm just showing you one here in these violin plots that are not present in the control setting. But surprisingly to us, at 15 months, these cells are no longer present. And that's not because this was a lower powered data set. It's actually exactly the same power at 7 months. It's just that we couldn't detect the cells there. And so one way we thought of interpreting this data was that perhaps these strategically located interferon-responsive cells were very important in the earliest stages of the pathology. And this was very similar if we look across a whole host of other genes that were marking these reactive astrocytes, other interferon-responsive genes like IFIP3 and STAT1, again, present early in the disease progression, but not later in progression. And this was also very similar to what Philip saw in some bulk sequencing data that showed a very early onset of these reactive subtype gene expression that peaked very rapidly and then also declined back to baseline very rapidly as well, following an acute inflammatory insult. So this is a little piece of data that shows that there's some neuroinflammatory states that are very lowly abundant and strategically located in the mouse brain. We call these interferon-responsive reactive astrocytes, or sort of humorously in the lab, we call these DUMAs. And we've actually investigated these also in the context of human postmortem tissue from patients with Alzheimer's disease. And similar to what we did in the mouse, we took advantage of great data sets that Liwei had generated and were able to show across studies that these cells are also present. But what we became really interested in over time is whether or not these cells would be localizing with pathology in the context of AD, and whether or not there were any other gene expression profiles associated with Alzheimer's disease pathology. So in this next vignette, I just want to tell you a little bit about a story that's available in bioRxiv. It will be out very shortly. And this was a collaboration between Philip Hazel in my lab and Emily Castranio, who's a postdoc in Michelle Ehrlich's lab up at Mount Sinai. And what they decided to do was investigate gene expression changes in wild-type mice and APPPS1 mice, which Tracy brought up earlier. And one of the things that I'm showing you here is just a UMAP, not of single-cell sequencing data, but actually of all of the spots from spatial transcriptomic data. And what Philip is asking here is, are there clusters of transcripts or clusters of regions in the brain that are associated with the disease pathology and not with the wild-type mouse? And so there's a couple of ways that we can investigate this. One is we can set up this sort of spiral diagram to show whether or not the genes that are expressed are similar across clusters. And in this instance, he identified 27 different clusters of genes. We don't need to go through all of them. This is just a means to an end. But what we can see is that there is definitely some clusters like this cluster 7 here, which is enriched for a whole host of genes. Around 20% or so are common across different clusters. You can see them connected by lines, but around 70 to 80% of them are specific to that individual cluster. So that doesn't tell us where it sits. Obviously, we need to be able to visualize where it is. And luckily, we did this with spatial transcriptomics. So what Philip can then do is take those clusters of genes and using this base plus module probing, he can say, well, where do those clusters of cells exist? And cluster 7 looked like it had a lot of specific clusters to it. It wasn't our plaque-specific cluster. It seemed to be present in white matter tracts in the brain. And we can go through and we can probe for all sorts of different clusters, and we can see a gene expression signature which is very localized to the hippocampus, or to the deeper cortical layers, or to the outer cortical layers. But again, none of these are really getting us towards a pathology-associated gene expression signature. But one of those clusters definitely did, and this is a cluster here we're showing in these UMAPs, just this feature plot showing that they are specifically located in the APPS1 mice and not in the wild-type mice. And so we sort of thought this actually could be our plaque-associated genes. And this is very similar to some work that Bart de Stroeper's group had done a few years ago, in which they called these plaque-induced gene signature, or PIGS. And so we decided to see if this particular cluster were these PIGS that he had described previously. So the first way we can do this is we can take our spatial transcriptomic data and we can probe back in with a module at low resolution and say, does it look like it's in the right regions? And indeed it does. We can see some signature in the hippocampus and in the cortex, but not in the subcortical layers. If we squint, we can see that it is a little punctate, but we can use our resolution enhancement tricks here, using both a module for what we're calling now these new PIGs and using the resolution enhancement of base. And we can see now we have these beautiful hotspots of gene expression changing once again in the cortex and hippocampus, but not in the subcortical level. But of course, this still doesn't tell us if this is distinctly associated with plaques. We're just inferring that. And so the way to really do this is to combine this with some staining for those plaques. And in this instance, Emily was actually staining for 6E10. And we can see wherever we have a positive stain for 6E10 plaques, we also have an overlay of this beautiful hexagon of gene expression from our base plus module probe. And every time we see these plaques, we can see we've got this nice gene expression profile for this plaque-induced gene module of these new PIGs. So we felt very confident that this expression profile was pathology localized and was in the right spot. And so we decided to say, well, is there anything else that localizes in these hotspots of that pathology? And so I have here an upset plot, which essentially is just showing us the number of shared genes in this vertical axis here, and the co-localization of these genes with a whole host of different subtypes of pathology-associated cells that have been described in the literature. So we can see down here, cluster 26, which is our new PIG module, is really clearly identified on its own as having a high number of genes that are very specific to it. But we can see there is some overlap with the original PIG cluster that was defined by Bart de Stroeper, but it just because of the resolution of the technology available at the time has a lower number of genes associated with it. But we can also see there is distinct overlap in these regions with things like disease-associated microglia, either defined early or late in pathology, with the interferon cluster that I talked to you about at the top of the talk. And what was really more compelling is we actually just look at the images of these. We can see that the pig cluster here that we've identified using our resolution enhancement with these hotspots around the pathology localizes beautifully with microglia that are dysfunctional in the context of disease, with these interferon reactive astrocytes. And we take this again as an important location at the interface between the periphery and the central nervous system, which of course we would expect to be occurring around these plaque pathologies. We also put in here another subtype of astrocytes that has been defined in the context of disease, these disease-associated astrocytes. And we found here that actually it wasn't localizing with the pathology at all. It was present broadly throughout the brain. And so we're interested in whether or not that was because these cells have a different location, or whether or not the methods that we used to define them simply didn't have the fidelity to localize substates of reactive astrocytes. So of course, now what we're really interested in, it's great for us to use this whole suite of genes. In this instance, there's nearly 400 differentially expressed genes in these new pigs. We have the capabilities to go and probe that back into this spatial transcriptomic data. But that's not what we would ever use in pathology samples. That's not what you would use in the clinic. You can't really make a PET ligand to hundreds of genes. And so we're interested about whether or not there was a single cell-specific non-amyloid marker of this pathology that we could use to understand what's happening at these cell-cell interactions. And so this is just showing us now, on the left-hand side, we have a wild-type mouse. We can see there's no hotspots with this pathology, which of course is pathology there. Here's our APPPS1 mouse with our hotspot with our new pigs. And so what Philip did is generated this plot here for us, which shows us on the outer ring here, the percentage of plaque-associated spots that are expressing a particular gene, low expression or no expression being dark blue, full expression or expression 100% of those spots being bright yellow. And on the inner surface, we're seeing the percentage of spots not associated with plaques expressing that same gene. And so what we can look for instance, in genes that have been associated with plaques before, things like TYRA-BP, yes, it's definitely enriched in 100% of those plaques, but it's also expressed in around 60 to 80% of the spots not associated with the plaque. The same is true for TREM2 or GFAP, things like this. But one gene that really stood out to us was this marker CST7, which was enriched in 100% of the spots associated with the plaque and was not present in any of the plaque, non-plaque associated spots. And so here I'm just showing you an image now of a single probe identification on this spatial transcriptomics that shows that these hotspots of plaques of pigs are exactly in the same spot if we use the single marker CST7. Now, of course, this is gene expression. It would also be really nice to see if any protein is associated here as well. And so Emily actually took advantage of, and I apologize, the resolution's a little low here, took advantage of staining some APPS1 mice with an antibody to CST7 that was developed and kindly gifted to us by Adriano Aguzzi. So here, if we just focus on the APPS1 mice, we can see that, of course, there are plaques in this hippocampus of this mouse, and there's some low hotspots of this CST7 marker. It's definitely there, it's just very lowly enriched. But we were also interested in whether or not we could augment the level of the CST7 and augment the function of these microglia. And so this was actually done by knocking out a gene IMPP5D using a CX3CR1 microglia-specific conditional knockdown. And we can actually see that this causes this CST7 marker of plaques to increase markedly, which was actually because there was a marked increase in the size and the number of these plaques throughout the brain as well. And just to show you this at the single plaque level, we can see in our control APPPS1 mice that don't have this SHIP1 or IMPP5D gene knockdown, we can see that, of course, the microglia are associated with these plaques, but they have low levels of this CST7 gene expressed. But in the context of knockdown of SHIP1, we can see that there is a marked increase in the amount of this plaque-specific marker in microglia that are associated with the amyloid. So I'm well and truly out of time, I think. So I just wanted to say that I didn't spend much time talking about the human-mouse comparisons here today. I just sort of mentioned it in passing. But we know that there is a number of reactive astrocyte substates that are evolutionarily conserved between rodents and humans. So Gil brought up the sort of neurotoxic subtype, as he's asking questions earlier today. We know that that is very evolutionarily conserved. We also know that this interferon responsive astrocyte subtype is also well conserved across species. We also know that these interferon responsive or DUMA reactive astrocytes aggregate in areas of peripheral immune cell infiltration into the brain, underneath the meninges and layer 1 of the cortex, around blood vessels. I showed you some staining from Brad Hyman's group. We also know it's present across a whole host of different diseases. I showed you data today for Alzheimer's disease. We've also seen in the literature and from recycling single cell data that's present in demyelinating lesions and also in acute injuries in the brain. We also know from some work that I didn't talk about, that Jeff Sadik and Michael O'Day also published a paper earlier this year showing that human AD-associated astrocytes really resemble these inflammation responsive astrocytes as well. We published this earlier in Neuron. And we know, as I showed you, these AD-associated astrocytes maintain these spatial locations as well, and some are localizing to plaque. So these interferon responsive astrocytes are localizing there, along with substates of reactive microglia as well. And then I also showed you that this novel reactive astrocyte pathology, which we had identified in inflammation in the mouse, seemed to also be associated with these hotspots of pathology, both in mouse models and also in human postmortem tissue. So, whoops, that was all I really had to say. I lost my acknowledgement slides going through, so I apologize for that. But I thanked all of the people as we went through, so I'm more than happy to take any questions that you may have, and happy to talk about the mouse models or the methods offline if anyone is interested further.
Thank you for a beautiful talk, Shing. So it's really your state-of-art use of the spatial transcriptomics. I admire that. So we have a few questions. So first question from Bob Weinberg. Are you aware of any transcriptomic profiling or heterogeneity of reactive astrocytes in other neuroinflammatory conditions such as Huntington's disease or Parkinson's disease? Yeah, that's a really, really good question. So there's a couple of problems with capturing astrocytes for single cell sequencing that I didn't talk about, but they seem to be largely excluded by the sort of microfluidic gel-based techniques that we are all using. So I think that in the broad scope of single cell sequencing studies that are out there, if you don't decide to study astrocytes, you end up underpowering your data sets, and so you can't tease out these lowly abundant substates like these interferon substates that are really interesting. So there are a number of really beautifully high quality data sets that have been generated from some of these. So there's a Huntington's data set generated by a group up at Columbia. Frank Quintana has generated some beautiful EAE and MS data sets up in Boston. And while those data sets haven't been able to identify these substates that we've been talking about today on their own, because of the low numbers of astrocytes, if we combine them and integrate them with larger power data sets, or just integrate across lots of data sets to increase the numbers, we can see that these substates are present. And what's remarkable, if you actually combine Alzheimer's data sets and Parkinson's data sets and Huntington's data sets and MS data sets, there is of course some changes that are disease specific, but a lot of these inflammatory responses are common across those diseases that suggest that while there is heterogeneity within a disease, there is some degree of homogeneity across diseases, which I think is really compelling from a drug development standpoint. Thank you. I think, Emily, if you have a quick question, I think we can squeeze that in right before our break.
Yeah, we'll get one fast one. I'm curious. I really like this concept that you have of these, multiple subtypes, which of course now all have these really interesting names. You've got dams and pigs, and but the functionally, do you? I just want to hear your sort of guesses on this. Do you expect that functionally these have different nuanced roles? You have these subtypes that all have some sort of inflammatory something that they're doing. What do you guess the different? Are they going to be different in what they're functionally doing?
Emily, this is the most important question, close and near and dear to my heart. So you're completely right. Transcriptomically identifying cells is really easy. Even anyone who doesn't have machinery in their lab can give a tissue sample to their core and generate transcriptomic data. That's reasonably straightforward. What is complicated is then working out what the function is. And this is where the major bottleneck is in the field. I see Li Wei nodding because we've had this discussion many times, and you can talk to anyone in the microglia field. Beth and I were talking about this just last week. So we have, one of the strengths of what we do in our lab as cell biologists is work out ways of recapitulating that gene expression transcriptomic profile using high quality in vitro platforms. And so what we tend to do is work out what is the cocktail of small molecules, or what is the transcription factors that would need to be turned on or off to drive the transcriptomic signature of those reactive substrates that we're seeing in our in vivo studies. So we have the ability to make in vitro reactive astrocytes that are neurotoxic, these interferon responsive astrocytes, and multiple subtypes of scar-forming reactive astrocytes as well, that recapitulate transcriptomically what's going on and are evolutionarily conserved both in rodent cells and in human iPSC-derived astrocytes as well. So there's 4 that we can play with at the moment. Those are the ones that we're investigating functionally. And I mentioned this morning to Nancy that we are really interested now in whether or not that function has a net positive or negative effect at different stages of disease. So, for instance, these interferon reactive astrocytes are not toxic to neurons like the neurotoxic astrocytes. When they become reactive, they require some of the same cocktails, like Gil brought up, interleukin and the TNF, they require that, but the addition of a different suite of small molecules to those as well causes them not to become neurotoxic, but they do still lose some of the normal astrocytic functions. And so the question is, are they actually really important in the early stages of pathology and sort of keeping the peripheral inflammation at bay? And then that function breaks down at some stage when you get to later pathology, and then those cells are no longer present, or do they die, or does something else happen? And so I think this is a really compelling avenue for research that we're sort of slowly investigating now. So watch this space. Great question. I love that question.